So how do they make the light? Well, it's not very dark in here, but I'll try. Because the rule is that all bioluminescence is chemiluminescence. I think you'll see it in this light. All we have to do is to mix the chemicals. If I add a fluorescent substance, I can change the colour. Is that dark enough to see that? Now, so one of the things that we've got to be clear about is that bioluminescence is not the same as fluorescence. Now, I said earlier that I want to excite you by exciting your electrons. Both fluorescence and bioluminescence involve molecules where their electrons have been excited. And the electron loses its energy, it emits a photon. But for fluorescence, you have to shine light. Can you see that fluorescence there? You have to shine light if you move the light. So if you're driving your car at night and you come across a cyclist, their fluorescent jacket or pants shine up. Once the headlight's gone, there's no light. But with the glowworm, it's still glowing without any light. Now this demonstration also explains why you don't get sunburnt indoors. Because this actually is ultraviolet light, and if you use window glass, it's opaque to ultraviolet light. But if I use a quartz crystal, you can see that it's still fluorescent, because quartz is transparent to ultraviolet light. And that turned out to be very important, actually, in business, people that sell microscopes. Because if you want to use ultraviolet light in a microscope, you really need quartz lenses. They're very expensive, sometimes £2,000 each. But if you don't use quartz, a lot of the light is absorbed by the silica glass. Yes, so I think I've got the demonstration. Here's a video. And if you add to that a fluorescent molecule, you change the colour. And this effect created $1 billion market for the luminous jellyfish. So the generic basis of the biochemistry of this is that you need a small organic molecule which is oxidized by oxygen and it's catalyzed by a protein. So it's an enzyme. This is a generic term which is confusing because each group of animals has a different luciferin and each individual species has a different sequence. But we showed that the firefly is 80% identical to the British glowworm, but it's completely different, totally different from the protein that makes light in a luminous jellyfish. Now, there's another important principle here. When you teach students about ATP, I'd be interested to know what your curriculum tells you to teach. Do you teach students that it has an energy-rich bond? Is that how do you teach about ATP? The molecule of life. Because there are textbooks, student textbooks, used in schools that tell you that ATP has an energy-rich bond. It's one of the great myths of biochemistry. It is not true, absolutely wrong. And one of the ways to demonstrate this with bioluminescence is if you put a glowworm on the floor and stamp on it, the light goes out. Now, this may not seem a surprise to most people, but to a biochemist, it's a big problem. Because you've mixed all the chemicals, why isn't it still making light? And an American, who's dead now, called Bill McElroy, thought, I know, it's because you've lost the energy. 1947, the Americans still think ATP has an energy-rich bond. It's in 2 or 3 textbooks. It's on a Harvard website. It's utterly and completely wrong. And we've got to do something about it because it's in the curriculum. I've seen it in books. I've seen it in revision books used in Wales by the Welsh Joint Board. It is totally wrong. ATP does not have an energy-rich bond. And you can see that why it doesn't do it here, because to make a visible photon you need this amount of kilocalories or joules. ATP has less than 10. So it can only make a tenth or a fifth of a photon. The energy comes from oxidation. It's burning without fire, so it's burning, but all the energy, instead of making heat like it does in a fire, is making light. The reason why ATP works to drive all the reactions in our body is because the cells keep the ATP well on the side of away from equilibrium. So it's a long way from the equilibrium, so that is why it drives. It's a potential energy, not because it has an energy-rich bond. Sorry. I'm sorry, yes, you'll be, okay, ATP stands for adenosine triphosphate. It's the molecule that drives all the reactions in your body. So when you eat food, the food, one of the things it's converted into, it makes ATP. I'm sorry, I the other teachers I do. So it's a potential energy. So when you make proteins, when you make DNA, when you make fat, when you store food, your electrical excitation, your brain, your heart, all of these are using ATP as an energy source. There's another system which is in the jellyfish, which is called a photoprotein, where they couple together as one. And all you have to do is to add calcium to make it flash. There's a whole range of luciferins. I just showed this to show the diversity of structures in these different animals. And one of the things I discovered was that this molecule is the most common chemistry causing bioluminescence in the seeds in all these different animals. So these are the phyla, these are the common names. So it's a very important part of the food chain in the deep sea. Now, the reason I actually started working on this was because I wanted to do a rather odd thing. I wanted to measure calcium. Well, everybody knows calcium is involved in bones and teeth, but what is less well known is it's a major signal in all the cells in your body. Every time your heart beats, there's a little puff of calcium which tells the cell to beat. When you do that, calcium goes up in the cell, causing the cell to contract, and then you can relax when the calcium is removed. If evolution has selected zinc, you could do that, but you couldn't do that. It would be very, very slow to relax. And the key experiment to show this was to measure the calcium in a live cell. Because this jellyfish, or hydroid, which is the part of the life cycle of a jellyfish, what I showed was that it flashes when it binds calcium. So by putting this protein into a live cell, the light that came out, we could measure calcium and show it was a switch. But it also gave me another idea. A flash is better than a glow. If you condense all your photons into a very small time, it's much more sensitive than if you spread it out. And what I discovered was that you can detect this protein down to 10 to the minus 19 of a gram. Whereas something like radioactive iodine, you can only detect down to 10 to the minus 16 of a gram. Why should I bother to worry about iodine? Well, we're at last getting to why I was brought to Cardiff, to develop new diagnostic methods. Because one of the methods that's used all over the world to measure things in blood uses antibodies. Antibodies are specific for things. So you could measure insulin, you could measure thyroid hormones, you could measure drugs, you could measure bacterial toxins, bacterial metabolites, bacterial proteins, cancer proteins. And you can also, using radioactivity, detect DNA, or RNA. You can detect whether somebody has HIV. But it was obvious to me in the 70s we needed to get rid of radioactivity. It's hazardous, it decays, it damages the molecules to which it's attached. Because of this sensitivity, I said, why don't we use chemiluminescence? A lot of people, including the government body, which were responsible, said, no, nobody's got a luminometer. I said, look, I built one, which is the most sensitive in the world, and that's why it works. Oh, but nobody else has got one. I said, look, these are billion-dollar markets. They'll make one. Well, we patented it. It was licensed to 2 American companies. The biggest machine in our clinical biochemistry laboratory in Cardiff is the machine that measures the light of this process.